Hello, this is Dr. Subramaniam Bangala, CEO of Reagent Biosciences in Bangalore. Yeah, I've had nearly 30 years of pharmaceutical industry experience, especially in drug discovery and drug development. Uh, the experience I had is primarily on pharmacology, toxicology, et- etc., which and also clinical pharmacology. I spent quite a bit of time studying how drug-drug interactions are related to toxicity interactions. Actually, these are all fatal interactions that cause deaths. In the context of COVID-19 today, I am seeing doctors using several drugs, maybe 10 to 12 or even more, depending on uh, the patient status, whether they are early disease or moderate disease or a severe disease. Yes, it's true. But the problem is when I noticed, even ICMR, the guidance, recommends the use of some of the outdated drugs like hydroxychloroquine and ivermectin. I am appalled, really appalled, because these drugs were not discovered in India, of course, we know that. Even hydroxychloroquine was discovered and approved by FDA in 1974 for uh, malaria and etc. So it's also extended to lupus because lupus is an immune disease and hydroxychloroquine appears to have some immunomodulatory effects. That's perfect and fair. When it comes to use of hydroxychloroquine today for COVID-19 disease, it's not administered alone. It's administered with many other drugs. Ivermectin, steroids, pantoprazole, Montelukast, you name it. And the antibiotics, doxycycline, azithromycin, etc. Now, why FDA didn't want hydroxychloroquine? They've already put out in July 2020 saying that we don't recommend hydroxychloroquine. There's no evidence that it works against the COVID-19 disease, and at the same time, it has potential liabilities for drug-drug interactions causing liver toxicity or even sudden heart attacks, arrhythmias in patients who are consuming it. And there is precedence hydroxychloroquine and azithromycin are primarily metabolized by the same enzyme, and so that's a concern. So these type of studies today are done regularly in pharma companies in the US to ensure there are no clinical liabilities before we move a new drug into the clinic. This is well established process. Drug-drug interactions are of great concern. There are at least a dozen molecules in the 80s and 90s were withdrawn because of their drug-drug interaction potential, causing fatal liver injuries as well as the heart attacks. So, why are we using a combination of 10 to 12 drugs? It's surprising. The ivermectin, on the other hand, also has no evidence. People presented in vitro evidence saying that, yeah, it is an antiviral drug. But when you go and look at it, first of all, they used Vero kidney cells, which are derived from monkey. And they don't have the same mechanism of uptake of the coronavirus. Apparently, they use a cathepsin gene, not ACE2 receptors. So, using that antiviral data obtained in monkey-derived kidney cells is not relevant to humans, because humans take up the drug with ACE2 receptors. It's a fundamental difference. So that antiviral activity is not justified to take into the clinical trials. Now you go back and say, okay, yeah, it has some antiviral activity. When you actually translate the in vitro drug concentrations that cause the cytotoxicity and antiviral activity in these kidney cells of monkey, they're about 5 times the concentrations that we see at a standard dose of 12 milligrams in humans. So, 12 milligrams is not going to produce those concentrations that are required to kill the viral cells. You need minimum 60 milligrams or higher dose. We don't have any clinical data at those doses whether the drug is going to be safe or not. And using a 12 milligrams and say, hey, we have dosed it 5 days and we have accomplished some mortality reduction, and they, most importantly, RT-PCR negative data by 14 days. 2 weeks is the quarantine time for people who don't receive any drugs, right? Why are we giving ivermectin and saying that it reduces the RT-PCR negative data, coming to negative status in 14 days? It doesn't make any sense. There were about 18 clinical trials, or even more, I think probably more than 25 today. I stopped counting the clinical trials. If you have to take 25 clinical trials to prove whether a drug is working or not, pharmaceutical companies don't do that. Yes or no answer, you need to get a effect in one clinical trial, right? Now maybe second clinical trial design much better in terms of the randomized placebo clinical trials, right? So it doesn't make any sense to use ivermectin and pushing it beyond the limits when it doesn't work. And also FDA noted in their refusal to authorize the ivermectin that they have data that several people took ivermectin as a prophylactic, and that includes hydroxychloroquine too, and they ended up in hospitals. These are not COVID disease patients. These are normal individuals. They thought these drugs could be prophylactic and took and ended up in hospitals. FDA has that data. We don't have any such data for the ICMR to present. We don't know. And in fact, a few doctors in India also asked me, what do you think? I said, yes, that's true, because these drugs do cause potential toxicities. And if you take hydroxychloroquine, right, it has 22 to 25 days half-life. One single dose of 400 milligram can last in your blood circulation maybe for 6 months or even more. So, drug is not getting cleared rapidly. A drug with 22 days half-life, if you give multiple doses, day after day, it doesn't make sense. Doctors should understand the pharmacokinetics. They don't even pay attention to what is pharmacokinetics. The half-life of the drug is very important. You just don't dose a drug that has 22 days of half-life every day for 5 days. It's ridiculous. So anyway, come back to issues with these ivermectin and hydroxychloroquine. There is no evidence they work, and they also have potential drug-drug interaction liabilities and other liabilities that can cause fatal interactions. So be careful when you prescribe these drugs to the patients. You don't need to. I know in the US and Europe, they don't prescribe these two drugs, and the patients survive. A lot of people do come out of the infection naturally, right? Our body is equipped with the immune system. 95 to 98% of the people come out of the infection. Natural immunity protects them. That brings me to the second question, use of steroids. Steroids are immunosuppressive agents. It's well documented in the literature. Yes, mild pneumonias like flu virus, maybe you can use them at low doses. But here, COVID disease has a severe cytokine storm. That is, trying to innate immune system is activated to stop the virus replication. Right? And what you're doing with the steroid is suppressing the immune system, and viral replication does continue. And the patient morbidity increases and he gets into more serious stages of disease. In fact, if you go back and look at the recent literature, or even back in 2020, in June, Italians published, they were surprised that COVID lungs, people who died of COVID disease, have extensive blood clots. It's called black lungs. I actually posted COVID black lungs, a small PowerPoint article showing that what COVID black lungs are. So, it's not disease of cytokine storm. You're treating wrong disease. COVID-19 is a vascular disease, and very recently there are few publications came out showing that spike protein can damage the mitochondria in endothelial cells. That is now going to be affected by steroids, ivermectin, and hydroxychloroquine. This is a direct effect of spike protein in mitochondria damage. That leads to a series of cascade reactions, which precipitate into blood clots. So platelets are activated, blood leaks are happening, and in the vessels in the lung, platelets are activated, and the microvessel suspension or clotting. At that stage, however much oxygenation you do for the patient is not going to come out of it. This is where the issue is that we are over-medicating the patients. Why do we need to over-medicate? On top of it, you're also giving your pantoprazole, Montelukast. These are all standard protocols. You don't have to have even allergic symptoms. These are prescribed routinely. And very surprisingly, I've seen some prescriptions where arsenica album, a homeopathy medicine, was prescribed by allopathic doctors, along with all these cocktail of drugs. It surprises me. So, I am not blaming anybody here, but we need to come to the grounds and understand drug-drug interactions are a very serious problem. So you're giving steroids, right? They are immunosuppressant agents. What do? They actually downregulate the cytochromes P450s in the liver, which are very critical for the metabolism of ivermectin and hydroxychloroquine or even pantoprazole, maybe levocetirizine, I don't know. But all these drugs do have metabolism. And what you're doing is further downregulating these drugs' metabolism, and you're actually increasing their concentrations to a toxic level, which they immediately attack liver or heart or the primary organs. Sometimes even kidneys may be included in the action. So, my friends, doctors, please don't use these drugs to treat patients. In my opinion, personally, when I'm looking at so many people next of kin from all my friends who are posting that, hey, somebody died today, somebody had massive heart attack, respiratory disease, it's giving heart attacks. Why? There's no oxygen to pump into the heart. Completely blocked. The lung is blocked completely. So there's no oxygen transport to heart. That's one thing. And the other thing is that the QT prolongation, which is through the drug-drug interactions, the azithromycin or hydroxychloroquine could go to a concentration that can inhibit the potassium channels in the heart that are required for heartbeat. Please read. There's a lot of literature out there that's changing every day. And 1970 discovered hydroxychloroquine should not be the drug today of choice when we have gone ways, miles and miles ahead of science. Today's science tells us that these drugs cannot be co-administered with other drugs. Ivermectin is a horse drug. We don't have enough clinical data to dose it to 100 milligram and you use only 12 milligram, maybe it's not sufficient. We don't have enough research data. Doing 30 clinical trials on something we really don't have a research data is very appalling to me. Yes, Dr. Corey is a pulmonologist, a career pulmonologist. He treated more pneumonia cases of flu, but he didn't know what the molecular pathology of COVID virus. Right? Coronavirus has a different molecular pathology. It is not flu virus. Unfortunately, this is what is killing the patients. Drugs, too many drugs are given. Please consult pharma industries. Merck said there is no evidence that ivermectin works. They make ivermectin. They should be profiteering with the ivermectin. But instead, they choose to do research and they think there's another drug called molnupiravir, maybe a better drug, as an antiviral drug to actually attack the virus and kill the virus. I'm happy that Zydus Cadila actually is importing PEG interferon alpha 2B. That's a better choice than any of these drugs doctors are currently using. Because it's an antiviral drug. It kills the virus. And it may also boost the antibody production against the virus. Giving steroids, you are killing the antibody production. You are increasing the viral replication rate. Doesn't make sense. Please, even for ICMR, I'm requesting, please change the guidelines. These are outdated drugs, ivermectin and hydroxychloroquine. Okay. And also, the steroid use has to be minimized as low as possible. Budesonide doesn't have any effect. A lot of studies are saying that it doesn't work. Yet, there is a craze Yeah, let's come to remdesivir, right? Remdesivir was a drug discovered for Ebola virus by Gilead. The clinical trials for Ebola failed with remdesivir. What makes us think that remdesivir is going to work for coronavirus when it failed originally for Ebola trials? Okay, that's fine. There were clinical trials done on remdesivir too. There was no effect. So I see everybody crazily going into the black market and trying to get remdesivir. Tisolizumab, I think that's a IL-6 inhibitor. But here, this cytokine storm is more than IL-6. As I said, it's a vascular disease. It's killing the mitochondria. The spike protein itself killing the mitochondria. So you cannot stop killing the mitochondria in endothelial cells by giving an IL-6 inhibitor, right? It's not mechanistically impossible how an IL-6 inhibitor can cure corona. Yeah, you can make the inflammation a little milder, but you're not actually stopping coronavirus spike protein, inhibiting or actually damaging the mitochondria. So we need to take a look back, need to change our guidances, the way we are thinking right now. I'm pretty sure most of the deaths that are happening today are not COVID disease related. They are more of over medication and due to drug-drug interactions. Thank you everyone. I really appreciate for your time and spread the message. I'm saying this is not the way to treat patients. I'm really surprised and we need to improve our basic medical technologies in India. Thank you. Bye-bye.